Fighting malaria with bacteria. Malaria is a vector disease, meaning that it is not passed directly between people, but rather through mosquitoes that bite people and animals. Malaria is caused by a group of single-celled eukaryotes called Plasmodium. Half of the life cycle of this Plasmodium occurs in the mosquito, and in fact, scientists found that it is here in the mosquito that this Plasmodium is at its weakest state. Biol and others at Johns Hopkins University altered the DNA of a bacterium that occurs naturally in the midgut of the mosquito. This genetically modified bacterium was made to synthesize a number of different proteins that attack Plasmodium in the mosquito.
The available methods for preventing malaria include pesticides and vaccines. However, the vector mosquitoes are becoming resistant to these methods. That is why there is an ongoing search to find ways to prevent and fight this disease. One new idea to prevent malaria is transgenesis, which involves modifying the genome of the actual mosquito. Although this method has worked in the lab, its success in the field looks slim due to the isolation of the mosquito populations, which prevents gene flow. That is why Dr. Wang and fellow researchers at Johns Hopkins have decided to experiment the use of paratransgenesis to combat the malaria parasite. This technique involves the modifying of the genome of a bacterium, P. agglomerans in their experiment, to secrete proteins or effector molecules that would attack the malaria parasite at its most vulnerable stage in the mosquito midgut, specifically during the formation of oocysts and malarial spores.
Let's take a closer look at the life cycle of malaria. When a mosquito carrying malaria bites a human, Plasmodium spores are passed on and enter the bloodstream. These spores make their way first to the liver where they multiply. Later they re-enter the bloodstream in greater numbers, and if an uninfected mosquito bites this individual, it takes up these newly formed Plasmodium cells. In a single blood meal, a mosquito can ingest as many as 10,000 Plasmodium cells called gametocytes. As many as 1,000 cells called eukaryotes are then formed, and they make their way to the mosquito midgut. It's in the midgut that these eukaryotes transform into little Plasmodium spore factories called oocysts. One oocyst can produce 1,000 new spores, but from the 10,000 original cells ingested, a single mosquito only has an average of 5 oocysts lodged in its midgut region. This oocyst bottleneck is where these scientists have employed their genetically modified bacteria.
After a Plasmodium-rich blood meal, the number of genetically modified bacteria in the midgut of a mosquito skyrockets to 200 times its typical level, allowing the surge of bacteria to defend against the invading Plasmodium. In the experiment, researchers fed mosquitoes a Plasmodium-rich blood meal and monitored the number of oocysts in their midgut 8 days later. Mosquitoes with effector molecules had significantly lower numbers of them.
The two most efficient effector molecules out of the 6 that were tested were scorpine, which comes from scorpion venom, and EPIP4. Each of these molecules decreased the average number of oocysts by approximately 98%. The others reduced the number of oocysts by 85 to 94%. And as a result of this decreased number of oocysts, the total number of infected mosquitoes decreased from 90% to 14%. However, to make sure that the production of anti-malarial proteins was not a great cost to the mosquito, the effect of the secreted proteins on uninfected mosquitoes was determined, and it was seen that the production of effector molecules had negligible impact on the lifespan of the mosquito.
P. agglomerans is an ideal bacterium to fight Plasmodium because it targets the weakest link in the Plasmodium life cycle, reproduction in the midgut. It is also found on plant surfaces and blossoms, making it easier to collect than other bacteria. Since the bacterium is found in the midgut of all 400 species of mosquitoes, it can be used as a universal tool to fight malaria by targeting Plasmodium reproduction in the 100 species that can spread malaria. Malaria is a problem at low latitudes across the globe, but the majority of the 1.2 million malaria deaths per year occur in Africa.
Implementation of this modified bacteria is challenging because of several political and ethical hurdles, one being that not everyone agrees on the utilization of genetically modified organisms. However, this method has proved successful in the lab with its ultimate goal of lowering Plasmodium levels and decreasing outbreaks of malaria. Another problem with the implementation is the baiting system the scientists designed. This would consist of cotton swabs soaked in the bacteria and suspended in sucrose solution. The baiting stations would be placed in areas where malaria is the most prevalent. This system has not been tested in the wild, so the outcomes are unknown.
So in conclusion, let's recap on 3 main points. First, the oocyst bottleneck. The scientists designed a strategy that attacks a malaria parasite at the most vulnerable stage in its life cycle. This strategy was proven effective in the lab. Mosquito infection rates from drinking contaminated blood dropped by 85 to 98%. Issues with this experiment are, will the new bacteria be able to compete with other bacteria in the gut given their new malaria-fighting duties? Can the baiting stations used reach a significant percentage of the population? Also, how do you sell introduction of genetically modified bacteria or mosquitoes into the environment? Here are our resources. Here is our scorpion. Our take-home is bacteria plus scorpion venom equals death to malaria.